welcome to today's webinar, where we're going to learn how our customer, Japan Tobacco International, is using KNIME for their toxicological risk assessment. Before we jump right into it, I would actually first start to cover some of the housekeeping for today. So you're using our streaming platform, and there are two tabs on your right side. The first one is the chat, so please use that one to interact with other people. For example, say where you're from, give some comments. But if you do have questions for us here in the studio or also about KNIME Analytics platform, we would ask you to use the Q&A tab, which is more right one. We have the people in the background covering the questions either directly or forwarding them to us in the studio so we can cover them later and also direct them to the speakers. If you have problems with the streaming, so the streaming platform works best with Chrome or Firefox web browser. If that doesn't work, of course, please try to refresh your browser. Of course, also try to check your internet connection. If that still doesn't help, we have seen that checking your cookie settings is sometimes a problem. You can also just try and go and try the private browser mode. And if you get completely lost on the streaming platform, on the left side of your screen, there is this stage button, and this will always bring you back to the screen. And with that, I want to welcome today's guests. I'm really super happy to have them here in our studio in Constance with us. So not going with the usual Zoom setup and having just people on the screen, but really having people here live in the studio is super nice. So welcome, Irene Abraham and Daniel Mucs. I'm super pleased you're here. Thanks for coming.
Here.
Thank you.
Before we dive right into it, maybe, and I think you have a slide on that, but just really nice for the people to get to know you. Would you maybe give a little bit of a background, where you're coming from, what was your training, about that? Maybe Irene, go first.
Yes. So I'm a chemically trained toxicologist. So I did some training in undergrad in chemistry, then decided I wanted a bit more biology. Ended up in toxicology, working on DNA adducts and different things like that before I moved into risk assessment, and then eventually the risk assessment at this nonprofit consulting company where they taught me risk assessment, brought me into JTI. And I've been there for 10 years now.
It's quite a long time.
Yeah.
How about you, Daniel?
Yeah, so hi everyone, again, pleasure to be here. So I have a background in chemistry, more the organic and analytical side as well, but moved towards computational chemistry from quite early on in my career. However, that sort of turned into computational toxicology, I guess, for the past 10 years, ranging from machine learning to supporting preclinical, clinical studies, doing virtual human modeling, PBPK, PBTK. Also had a stint in academia as well, and joined Irene's team 3.5 years ago at JT International as a toxicology manager.
Thanks. So actually, Daniel and me, we know each other from when I was not with KNIME yet, and Daniel was not with JTI yet. But already back then with the EU project, you were working with KNIME. So what brought you to use KNIME back in the day? Because I think you're actually using it for quite a while now already.
Yes, that's correct. Sort of, yeah. We met in a previous slide, but also a few more previous slides before I sort of got involved with KNIME, which was turning 10 years ago, I guess. And already there, it was sort of great, the sort of low code or visual coding aspect of it as well. And also the chem informatics as a chemist, that's what I've been sort of looking for, to find that, to be able to handle molecules, to be able to link data, sort of have a trace of how you did things rather than just copy pasting different Excel sheets or SDF files and so on. So certainly it has been a great tool for over a decade to pretty much anything I've been doing so far.
Cool, nice. And with that, I would hand over right to Irene, and let's hear about some toxicology. Looking forward to that.
Thank you. So I'll take you a bit through how we've been using KNIME in toxicological risk assessment at JTI. So again, I'm Irene, and Daniel's here with me, and I lead a team of toxicologists where we would support reduced risk product assessment at JTI. So in general, I'll go through a quick introduction into toxicology and risk assessment. We'll go through some of a use case of where we've used KNIME, go through the challenges, solving some of the challenges, and then Daniel will pick up with some of the improvements, the lessons learned, and an outlook. So the tenet of toxicology is all things are poison and nothing is without poison, only the dose makes the poison. So this is kind of the grandfather of toxicology. Paracelsus says this. And so what's important to know is toxicology is extremely broad, it's extremely complex, and even things that you might not think of as being toxic can actually, at the right dose, cause harm. For instance, even something as simple as water. If you have too much water, it can harm a living system. And so that's just toxicology in a nutshell. But this is a combination of lots of different disciplines. You have ecotoxicology, you have more focused on drugs, you have more focused on what we do, the tobacco products, and you can have many different areas, disciplines, sub-disciplines. So very broad, very complex. And because it is so broad and complex, you have data from many, many, many different sources, and they can be very different. And you're constantly looking from place to place to put together a story, to tell the story about what is actually happening in a living system. And add on top of this, things that we need to know about the chemistry of it all. So chemistry can be simpler because a reaction happens, and it happens this way when you do certain things. When you add in biology to this, it gets all very complicated. Adding a living system makes it all very complicated. And so when you're considering all these different moving parts, toxicology is our bread and butter. But what else is going on? So in toxicology, we're gathering all this information for what purpose? We're gathering this information because we want to answer a question. We want to identify whether something is or is not hazardous. And then once we decide it might be hazardous, we then have to look at the exposure of it to see whether it's actually getting to a person or to whatever living system we're studying. And then also consider how much is there. And so this is what the tenets of risk assessment. And risk assessment is just one part of what we do in our company at JTI for product stewardship, to make sure that the products that we put on the market meet the intended use for the consumers. And so another thing to mention is you have the hazard that I talked about, you have exposure, but if you have no exposure, you can't actually have a risk. So it's all working together in the grand scheme of things. And the thing about working in our industry is that you're gathering all this information I talked about before. This is a very time-consuming thing. And you have substances that you're looking through, and these are in mixtures because you're not just looking at one thing at a time. So how do you go through all this information? And you have limited time to go through. People want their answers. They want them as quickly as you can get them because this is a business. So therefore, we needed to look at a way to have a high throughput of this information faster than what we were traditionally doing of gathering all the information, spending all the time to make sure we're looking at the full picture. And then once all this is gathered, making our analysis. But the data gathering step isn't actually what us as toxicologists should be working on. Data gathering really is something that we could automate if we had the right system to look through it. And so this is where KNIME came in for us. So then after you have the data gathering, you can spend more time on linking the substance information with the chemical side to the tox information, biological side, to make the decisions that you need to make. But for this to happen, the data has to be structured and visualized well. And because you need to know what you've done, who's done it, and when things may change, where it changed, and whether you need to change your assumptions or your conclusion, you need a solid audit trail so that you can go back and make sure that your answer today is still your answer tomorrow. And as we go forward, the field of risk assessment and toxicology is going into its next generation. It's called next generation risk assessment. You'll see things like other terms like NAMs, the alternative methods of risk assessment. And what we have here on the slide is showing the different parts that go into this. And so this is just to show you a bit more of the complexity that goes into the risk assessment as we do them today. You have the exposure estimations with all the different scenarios that could go into how the living system is exposed, how it's moving through the body, how it is going into the person. But then you also have gathering all that information. And when you're gathering all this information, you have to know about the structures, you have to look through the literature, and then we have a bit more of what you'll hear from Daniel about the in silico predictions for, in some cases, data gap filling, where you don't have all the studies to tell you what's happening, but you do have a pretty good idea of what's happening with things that may be similar. And so can we predict what's happening without needing to do something like maybe an animal study? Can we predict what's happening or use this prediction to guide us to doing more appropriate studies to limit the use of animals? And with that, I'll turn over to Daniel.
Brilliant. Thank you so much for the excellent intro, Irene. And let me just for a sec try to get the pointer here and actually pick up from what Irene mentioned. So here, in the next generation risk assessment paradigm here, so this figure is from a publication by Unilever Safety Assessment. Only one part is the in silico prediction here you see at the bottom. So it's a big puzzle, a big detective work, technically, what we have to do as part of any risk assessment to find all the different pieces of information that Irene mentioned. So here, again, taking a figure from another excellent publication on proposing in silico toxicology protocols. How can we combine, or what is one way to combine all the different pieces of evidence we have? So this means your experimental data, your statistical model results, your expert alerts, or your read-across results. Different data sources have different reliabilities. Not every model is the same. Not necessarily talking in silico models, whether we take in vitro systems, so things done in the test tube, or in vivo studies. But as Irene said as well, really we're looking at to use these methods to avoid having to do, for example, animal experiments in line with the 3R principles in terms of reducing, refining, and ultimately replacing these type of experiments. But still, historically, a lot of data is available. And as Irene mentioned as well, in silico methods are really sort of called upon when there are data gaps, because usually for many in silico methods, you pretty much just need molecular structures. But of course, using these methods, you can generate, again, a large amount of data. So somehow you need to assign some sort of reliability scores. Sometimes you have to do some sort of assessment. And to do this in the broad picture of combining scientific data and linking it to, say, adverse outcome pathways, various effects, mechanisms that we see here, and then combine them into specific endpoints, which ultimately feed into the risk assessment. So this, again, sort of shows how deep the rabbit hole is, how you can have literally thousands of data points for a single molecule. So this is here a Society of Toxicology poster from 2021 that Irene's team, and also in collaboration with another team down in Vienna, we presented at SOT. So here, the sort of case was a non-targeted chemical analysis, otherwise known as scanning chemical analysis, which means finding a lot of different molecules. So here, KNIME really did the heavy lifting. It did the extraction, transformation, and loading of all these different data sources, which included various different public data sources, QSAR models, so quantitative structure-activity relationship models based on chemical structures, structural alerts, and so on. And also to provide this in a digestible way. If you think of it, you have potentially hundreds of rows, hundreds of substances, potentially thousands of columns. That's just overwhelming to try and do science. You need somehow to condense the data, to summarize it and visualize it so that scientists can digest it. And if needed, you have the audit trail to be able to sort of go down deeper of how the data was combined, what were the rules, and how does the original raw data look like. So let's first set the picture for broader requirements where certain KNIME fits in. In general, for toxicology, as Irene mentioned as well, it's a lot of different disciplines. But really, we are interested in molecular structure. We're interested in the chemistry. So we need to be able to search by various different IDs and structures to be able to read across. Of course, we're going to touch on this, that when we talk about read across, it's not just chemical similarity, but biological similarity. Also, we need the structures for machine learning, deep learning prediction. So for QSARs, QSPRs, or for TTC, for threshold of toxicological concern, or for what is referred to as virtual human or virtual preclinical species modeling, so physiologically based pharmacokinetic or toxicokinetic predictions, reaction predictions. And again, as Irene touched on this, it's not just single molecules that we look at, but we actually look at multi-constituent substances as well, so mixtures. And then, of course, all this has to be linked to the biology. And this means any data, including literature, internal, external data, in vivo, in vitro, in silico, and so on. Now, searching the data is quite key as well. So again, KNIME has been really a useful tool here to be able to use various different identifiers or structure searching as well to get all the relevant tox data. And we do say it a lot, but the history, the audit trail availability is really, really a key feature. And then we need to be able to upload data for both chemistry and biology from various different sources, structured and unstructured. And all of this need to be integrated with existing IT components we have, meaning, for example, single sign-on, for user ease, SharePoint integration for unstructured data, OneDrive integration, potentially what data could be there. And one of the features actually we looked at when sort of constructing this ecosystem of computational tools was KNIME. As quite early on, it sort of really showed it to be an extremely useful tool for our purposes to do prototyping. And here would like to mention our KNIME partner, actually KNIME's partner here, so our partner as well, who helps to sort of make the prototypes be deployed and be used by other toxicologists. So Soluzione Informatica has been an excellent consultant for us in making sense of our prototypes and to optimize them for actual deployment. But overall, we did want to have a light system, and KNIME also sort of ticks this box. So something we talk a lot about science as well, and we already talked about visualization, but still, for me, the most important part is to have good visualization, to have an easy-to-use interface for non-IT expert end users. Most of our colleagues are excellent scientists, excellent toxicologists, but they are non-IT experts. So again, KNIME with the low-code, visual code approach has been provided an excellent tool to be able to every part of the toxicological risk assessment, to what Irene mentioned as well, to be able to perform the data gathering, to ultimately provide some simplified sort of visualization tool where decisions could be made, whether we have enough data, do we have the right data, what next steps should be taken. So here, I'm going to talk about some of the recent improvements that we have done using KNIME as well as part of the toxicological risk assessment process. The first one is the chemical identifier checker component. So a bit of a background info here, the problem we're looking at. So our problem is to be able to convert CAS registration numbers into small strings to be able to run later similarity search, which I touched upon already. The general idea was to use the NIH NCI Cactus Chemical Information Resolver web service. Now, the problem with this, even if you use KNIME directly, if there are non-valid identifiers, meaning containing illegal characters, the GET request node will fail. And that means if the calls that ran successfully before are going to be lost. So that means if you were running thousands of substances potentially, and it fails at the end, everything is lost. Therefore, the solution is a component that checks the validity and excludes values with, for example, white spaces, non-digit, incorrect total length, or identifiers with an incorrect check digit. So CAS, actually the last digit within every CAS number is a check digit. So there's a mathematical formula to check the validity of the CAS number. Now, there are some notes here, for example, with doing the check digits. So yes, probably it wouldn't be a huge issue here, and it wouldn't return a failed GET request. But if we can do these sort of checks and reduce the amount of calls that we do to the service, already would help sort of on the front-loading side. And please note that the demo we're going to show in the number of input rows have been truncated for this demo. So here you see an animation, and actually showing the input that you see here. So you see it's an ID, it's a CAS number, and its name. And already here, there are a few sort of problematic CAS numbers, which is sadly quite common with public data sources, and they come in various shapes and forms. And we'll see on the bottom branch here what happens if these CAS numbers are sort of fired to the Cactus service. So you do get sort of failed response, and yeah, so you sort of need to do some manual work. But with this identifier checker component, you can easily configure to select, okay, which is the CAS number, and then filter out these problematic rows. So here in the bottom bottom output of the row filter, you can see these problematic CAS numbers that have been identified. this is going to be filtered out, and it's only just the top branch, which is going to be submitted to the Cactus service. And that means at the end, you see there's no error here, so it's going to be executed behind. There we go. And it's all green, based on the KNIME traffic light system. And we do have the output. And where the system was able to translate the cast number into a smile string, we have the response. So taking on from this, why do we need such a component? So actually, we want to be able to do what we touched upon, read across. And that means a similarity search. Now here for the demo, we're actually going to look at the US EPA Toxicological Value Database, ToxValDB for short. And the goal here is to find a point of departure data of similar substances and be able to also use these custom similarity metrics. Because as I touched on it already, it's not just chemical similarity what we want to look at, and that's sort of your standard features that you get in currently as existing solutions, but you actually maybe want to look at biological similarity. So for this, as mentioned, using the US EPA ToxValDB that was published a few months ago, and the similarity metrics currently available in the US EPA ComTox dashboard, so you can do a similarity search there, but they do give some unexpected hits, and that sort of stemmed this project as well. Now, the problem and the solution. So for the similarity search, we need the smile string from cast numbers. This sort of links into the previous example that was demonstrated. Now, the data sheet itself came with almost 1 million rows, but many cast numbers were duplicates. Actually, out of this, there are around 32,000 individual cast numbers. And what happened from that, just looked at the UPAC names first and used the RDKit from UPAC node to be able to get the structures from these names. Now, where this conversion was not successful, actually looked at the corresponding cast number and did the cast number sort of checking as well that we previously talked about, run the GET request to Cactus, and then excluded any responses that didn't provide a chemical structure, so didn't provide a valid smile string. And pretty much what we found that around 24,000 of the 32,000 cast numbers could be converted to around 75%. Now, the data table was extended with the smile string if it was present, and it was deployed to a database to improve the search speed. And access and query to this database was done with KNIME for the similarity search. Bit of some technical info as well for those who are interested. So the database itself was a PostgreSDB with an RDKit cartridge, and right now deployed as a Docker image. We're also looking at in the process of setting up it in an Azure infrastructure. The molecular drawing tool is Catcher. It's open source, done by EPAM. And the available fingerprints currently, as much as I talked about biological fingerprints, for the sake of the demo, we used out-of-the-box Morgan fingerprints with radius 3 and max fingerprints. And also, we do note here what we found that by default, RDKit applies a threshold of 0.5 for similarity, so something to keep in mind if you use that type of similarity search. So again, this is how the workflow looks on the web portal. So what you're going to see here is to be able to put in a smile string. Of course, here, I guess you could use already the Cactus service too from another identifier to generate the structure. Then it shows up in the Catcher. You can modify it. Then you need to configure which fingerprint with molecular representation you're using. Also, there's 2 different similarity metrics included, so Tanimoto and DICE. And also need to do the configuration. Here, the threshold is put down to 0, and the number of nearest neighbors is going to be set to 6. And then just need to click Next. And then the molecules which fulfill the similarity criteria will show up. And also for toxicologists, now there will be the data already showing which type of point of departures are available, which are really critical in the dose response or risk characterization part. Now, for some molecules, there was a lot of different point of departures. So again, for the user, we made it available that they are able to subset here, for example, low ALs, but no ALs or any other that might be deemed sufficient for the risk assessment process. Now, even when that's done, you do get a lot of rows and you do get a lot of columns. So again, KNIME is great in this as well to be able to dynamically adjust the visualization so that the data is not overwhelming and key columns can be selected by the user on demand. So this feature has been added to the workflow as well. So here you see sort of a large number of columns. Subset can be interactively selected. And then once that's done, it pretty much visualizes straight away. But it's also captured in the workflow for the output. So when we go to the next step, pretty much a file name is automatically created. Here, for sake of audit trail, you see there's a molecular identifier with smiles. What was the similarity metric and other settings as well can be stored in the file name. And clicking next, the file can be downloaded in Excel format for further use. And that's going to be opened in just a second to sort of show you how it looks like in Excel. So there we go, pretty much what we've seen on the KNIME web portal. So just again, a bit of looking under the hood. This is the part where initially the unique cast numbers, unique entries were looked at. So as said, first, it was the RDKit from UPAC node on the top branch. So what was able to transform or find a molecular structure from the UPAC name was done. For anything else, the chemical identifier checker component was used based on the cast number, and then it was joined up or concatenated with all the other results. Now, the last improvement we're going to talk about is ToxTree integration in headless mode. So ToxTree is one of the computational toxicology tools based on various decision trees that have been used for quite a good few years now. And the goal was to integrate ToxTree into a KNIME workflow so it can be automated, therefore reducing the manual error or manual steps that a toxicologist needs to take to generate the input data and then also pipe back the output data. Now, the problem was that ToxTree can be integrated via command line only. We provided the link in the FAQ as well. The solution was that, okay, let's deploy ToxTree to the KNIME server and use the external tool node to run it. So this can be just pointed to the Java executable, insert a command line argument, and the column is automatically selected if it's called SMILE, so picking up the chemical structure. So again, how it looks like in a KNIME web portal deployment. Here, just need to provide an input file, which will be shown in a sec, which contains, in this case, I believe it's going to be name, cast number, and smiles. So here we'll see actually how the file looks like. There you go. So cast, name, and smiles were available, of course. So this is being uploaded into this KNIME workflow. Then click next. This data is loaded. And then one needs to select which decision tree should be used within ToxTree because it's a Java implementation and because how it was done was fairly easy to do this integration. So this way, again, the user doesn't need to know the software itself, but they get sort of the key features integrated within KNIME. And then click next, do the execution, and then they are presented with the data matrix here, which again contains a lot of different columns. So like we've seen in the previous example, one could add interactive column filtering options as well, or give ways to summarize columns or to download it in various different formats. So lessons learned at Outlook. All in all, KNIME has really provided some great features. As said, the visual coding is great for non-IT experts. It really helps with the black box thing, because of course people see, okay, we push a button and this happens, but coming from the scientific perspective, we need to have trust and understanding what is actually happening under the hood, so it's not a black box. And due to the visual coding nature, it is self-documenting, which is really nice, not having to necessarily straight away write heavy documentation. And the KNIME desktop analytics platform have been provided great for prototyping, as said, and then ultimately for deployment, the KNIME web portal graphical user interface with single sign-on is brilliant. And again, would like to mention here Soluzione Informatica, and they have really, really helped us to bridge this gap. Now, we did encounter some challenges along the way. Integration of KNIME with other systems does require significant IT efforts and skills. So please do keep this in mind. Also, troubleshooting is not always straightforward. For example, if you run jobs on the KNIME web portal, users by default probably won't untick the deletion once done. Therefore, if something went wrong, we don't really have a job to look at, and there's some significant log file dialing to be done. All in all, feedbacks from toxicologists is definitely positive. It is much better than copy-pasting from different data sources and tools, therefore reducing the human error. But as we touched on it already, there's a significant speed up on the initial data gathering as well for hazard identification, at least 50-fold that we have seen. Of course, the more data, the higher the speed up. And there's always questions on what else could we use KNIME for, so there's plenty of enthusiasm. So that was us. Thank you so much for your attention.
Thanks, Daniel, and thanks, Irene, for this really nice presentation. Before we dive into the questions that is coming from the audience, maybe a question for Daniel, because you're, as I understood, a bit more hands-on with your role at JTI. So how was that adopted by your team? How regular are you using it now?
We are using it on a daily basis. And I guess the adoption was literally sitting down in the same meeting room and building it piece by piece. And I guess the team saw there as well the benefit of it, and also see how, for example, NIME workflow can be constructed, and then it's nothing scary. And actually, it's a really beneficial tool. So
also your team is now building workflows?
With various degrees. I guess it comes to the prototyping thing. So building workflows is one part. How you deploy workflows in a reliable manner and in an optimal manner, that's a different story.
Okay. Maybe a question for Irene, because you're more of the manager role here. So how does that affect a little bit the team, or how your team works? So the processes, what are the beneficial outcomes from a manager perspective? What would you say?
Yeah, my perspective is it's getting us faster through the data gathering step. So it's giving the ability of my team to focus on doing the actual science, the actual analysis of the information, which is very, very beneficial.
Cool. You said that actually the data gathering step is the quite tedious one. Is it also when you would have a wish where the field of toxicology would go? Is that one of the biggest wishes you would have, where it should develop, that we're having better data sources, better REST APIs or API interfaces? What would you say?
So as a non-computational toxicologist that would benefit from this, yeah, the desire is to have as much of this information in a place where we can use it very efficiently, effectively, automating the parts that don't actually take the human brain to analyze to get an answer.
Yeah, so not rely on copy-pasting and querying web pages, right?
Yeah, and I guess to add to that, it's ontologies, and I guess it's the whole science field. The different data sources have different identifiers or different flags, different classifications. So again, it's good to have tools like KNIME, because at least you know how it was for the different data sources, because you will forget over time as well. But that would be nice if we wouldn't have to do so many column renames or column filters or joining with different options.
Maybe we have a question from the audience that goes roughly into that direction. So how would you group unknown chemicals if it doesn't have a UPAC name or a functional group?
Oh, that's a...
I think maybe the CAS is already helping there, because that one is really nicely allowing you to transfer identifier to identifier.
Yeah, I mean, well, you don't have a functional group. If you have a CAS, and even if it's mono multi-constituent substance, you can get a structure, right? So that's sort of my confusion here, because you will have a functional group. You can sort of do some similarities. You can do in silico methods to try to do, okay, even in QSAR models or PBTK models, do they behave similarly?
Okay. Then, of course, we have the question if the work that you have presented is available for the public. So is that all open source that you have shown here today?
Yes.
Or is it just in-house for you guys?
Well, currently how it is in-house, so the workflows are not available, but it's all open source. So everybody should be able to sort of reproduce it.
Yeah, yeah. Then we have another one that might have been already answered in the course of the webinar. To build the workflows and maintain them, are you guys doing it, and are you responsible for it, or do you have any KNIME developer in your team responsible for it?
Right, so primarily I'm taking the role within that in the team in terms of maintenance, but as mentioned, when it comes to production deployment, Solution Informatica is huge help. So again, one shouldn't underestimate the resource and effort required on having optimally working KNIME workflows as well. So prototypes are great. They're great for proof of concept, but when rolling out to the team, there are some other requirements as well.
They're a prototype, right? You will always stumble into something that you didn't think of. Yeah, yes. I know that very well. All right, then we have another one, another question. Which class devices is applicable for in silico methods? I am not exactly sure what the class devices. Does it say something?
Not sure. Maybe.
If not, that would be cool if you could specify that again in the Q&A. We will see that in a second here on the screen, and we would just jump to the next question. I am wondering if consensus can be made when different source prediction results are expected. If consensus can be made. When we have different source predictions and prediction results are expected, if you can have consensus on that.
Yeah, this is one of the difficulties of this area is even in normal toxicology, I'll call it non-computational, you put 15 of us in a room, you have 15 different answers. And getting consensus really involves having more people doing this type of work, gathering together and making the best practices. And right now the field is developing these best practices. So probably today, the consensus methods are still under discussion. They're not ready for us to say, yes, that's how you get there.
Yeah, I know that. It's like the EPA has a different standard than the Europeans, and again, a different standard than
Absolutely.
Japanese and so on. Oh, yeah. You have shown this big workflow, and that is also collecting data from different sources. How are you having these sources right now, and how are you maintaining them? So if there's new data being released by the US EPA or something, how do you make sure that this is really ending up at the toxicologists in this dashboard?
Right. So it depends, I guess, on the source. If you have an API, of course, that's the best way. If KNIME can connect seamlessly, that's the best. But many different data sources, public data sources that we use might just, have a link to an Excel file to download. I mean, the ToxFileDB is an Excel file, so we have to live with that, capture that somewhere locally, because what we have seen with public data sources as well, they disappear. And of course, for the audit trail, that's not really a healthy thing. So again, have it somewhere locally, link to it, or even if it disappears and it's an API, KNIME is great for that because you can save workflows, execute it. So the data will be there even if the original source might have disappeared.
That has also saved me already quite a few times,
yes.
Actually. Maybe because you also mentioned the audit trail, and KNIME by nature is already some documenting what happened to the data and so on. But have you ever gone through an official audit trail, and have you gotten feedback from officials on your on the trail you're keeping?
So far, no. We do internally go through our own QA, QC, making sure we can find information and that it doesn't get misplaced or lost. But actually, we right now would point to the example from the PMTA for PMI, where they use some of these techniques. And in their in silico techniques, the FDA was asking questions around the auditing of things. And we know that the FDA has been working and looking towards KNIME and talking about some of the solutions that KNIME has in this area.
Yep.
Yeah, so just to add on that, just because you have a tool, it doesn't automatically mean so you have to use it in the right way as well. That's an important aspect with any in silico methods. It's not just a push of a button. So you need to be explained how that button was designed, what's the principles behind it.
Yeah, and also thinking ahead, like what do I actually want to capture? What is the metadata that I want, and also FDA wants, and then designing accordingly. There are plenty more questions coming. So how are you dealing with confidential data with KNIME? Have you implemented company security rules or are you adopting in the website? Oh, I didn't get the last point. But yeah, so how are you dealing with the confidential data? And if you have any security rules?
Yes, we have security rules, but also the information that we're evaluating and running through KNIME doesn't go off to KNIME servers and get saved there. It's still kept internally. So all of our data restrictions and protections still apply. So there's not this potential for the data to leak out or to become non-confidential at any point in time.
So also the KNIME server, and I mean, it's not actually a server, right? It's just a software. So you have that on-prem within
our
your
own
network
infrastructure. Yeah, that's the thing. So, policies and so on do apply, as I guess, to everyone. Also, I guess the single sign-on here as well, is great because you sort of can link it. It's not sort of standalone users or groups, on the KNIME deployment.
Yeah, it's true. It's actually also a nice audit trail already within. Is it possible to parallelize jobs in KNIME when we automate the process with big data? So I think that is maybe going a bit more in the direction of performance? Can you parallelize that? And I admit that this is quite a techie question.
Almost like sort of back at KNIME, sort of this, how is the parallelization going, and elasticity. I guess what we say a lot in toxicology is, well, it depends. So yeah, at least in our experience.
Into in a situation where you felt like, oh wow, this is now slowing down because it has to crunch that much of a data or something?
Yes, of course. I mean, there's only so much resource a server has. So of course, if you increase it, but you have to think in mind being cost effective, you cannot just have for the sake of it, large hardware resources. So of course we ran into this, and then you have to go, okay, maybe I need to run the nodes in a certain order, or maybe wait on a specific branch. So yes, you can run it in parallel. Is it going to really help your case? Not necessarily.
Sometimes, yeah. Yeah, and this is also maybe going in the direction of chemical toolkits in the background. So that would be something. What's your favorite chemical toolkit that you're using in the back? Is that RDKit, because you showed a lot of stuff with RDKit?
Yes, it is RDKit. It's a historical thing for me. I do use sometimes CDK or others available as well, but RDKit is usually the first one that I reach for. Yeah,
and that is maybe a tip for speeding up things if you're converting between different toolkits, and the data has to convert in the background. That is also slowing things down. So once you are with your favorite toolkit, it's nice to stay there. Do you have any thoughts on how to incorporate exposure PBK into KNIME? That's quite a good one that we are also figuring out right now ourselves.
Yes, so there are, I guess, multiple different ways. One could use an R snippet node. We mentioned the US EPA. They have the high throughput toxicokinetics. So you can just take that off the shelf, or you can code your own, ordinary differential equation system in R, or in, for example, a Python node as well. But you can use, I guess, other commercial off-the-shelf tools as well. There it becomes a question of like, do you have an API? Can you pipe it similar to ToxTree? So short answer is yes, you can, but sort of the actual practicalities of it might be sort of a bit longer.
No. Do you use version control for the parameters of the workflow nodes?
So I guess, yeah, snapshots here, but yeah, version control, we do use sort of snapshots for everything that is possible through the KNIME server.
And you're also capturing the version of the data that goes in. So if that's an Excel sheet that was released, whatever, September this year, you will always know who ran the workflow and what was the underlying data at that time.
That's correct.
That's true. Okay. Besides toxicology use cases, what other OSHA case challenge problems would be worth deploying KNIME for machine learning and risk safety performance predictions and monitoring?
That's a lot there, and I'm not really sure.
So I think what the question is can we use the same thing to go through and use OSHA, so occupational risk assessments, and using it similarly? And we haven't really put that data into the system, so I'm not sure how easy it would be, but it is possible to do it, indeed, similar to what we're doing.
Yeah, it's
for the
possible.
other sources.
Why I was sort of thinking, because machine learning comes into that as well, and it's sort of the favorite buzzword with AI. People like to throw it on top of it. And I guess with toxicology, something we should remember that there's been guidance out, for example, the OECD QSER guidelines for coming to 20 years now. I mean, these methods, machine learning is nothing really new for computational chemistry. We're talking a good few decades. So yes, you can, I guess, incorporate any data you'd like. Yes, you can do machine learning. Is it going to be really meaningful and following these guidelines? Again, that's a whole different story.
Okay. Based on chemical characterization, can we leverage biological testing? In which case we can do that?
I don't know if I got the...
So if I understand the question
Yeah,
well, can we use biological data to help us characterize chemicals? Is that
right?
So if we have chemical characterization, can we, with that, can we leverage biological testing?
Okay. So the question is,
if so, in which case can we do that? If we have,
So to do something like this, if you have the chemical characterization, you still need some kind of biological data to link to, because just because a chemical looks like another chemical doesn't mean it's going to work that way. We even have examples of where something that's a isomer, one is very toxic and one is just fine. So it's not enough to simply look at the chemistry and be very sure that the biology is going to end up a certain way, but you can have an idea. And as a toxicologist, until you have better information, we usually operate according to the precautionary principle, which is, unless I have other information, no.
Yeah, I remember that. So is the front end of your tool KNIME itself? Can KNIME be configured to take inputs from other tools or front ends? And I think that is really nicely covering your story as well, right?
Yes, I guess that is the short and long answer is yes, it can.
Yeah, that is actually also something nice when it comes to Python. And I don't know if you guys are coding, but you already said most of the toxicologists are not quite that skilled with scripting and coding and so on, but KNIME can be a really nice front end for that. So you actually have the script in the back end. And that's just kind of, you give users the possibility through KNIME to give parameters and then insert them in the code, but have the nice front end of KNIME. So that's quite nice.
I mean, that's another thing with codes. Even if you do code, you have to think that you will need to explain every single line of code or every single bit potentially, especially when coming from the regulatory toxicology side. So you can code a lot, but you will need to explain. So again, even though I'm familiar with Python, familiar with R and other programming languages, I would try to use as much as possible sort of out-of-the-box nodes within KNIME rather than writing code.
Yeah, it's true, because they're really, it's even for somebody who doesn't know how to a for loop works or something, they can see exactly what's happening to the data, right?
Yeah, and I guess it's the unit testing feature as well. You can check exactly what happens in every single bit. Sorry.
And the other benefit is if it's something that's already in the system that we haven't coded ourselves, then it's reproducible for other people that are trying to check the work, that are trying to reproduce what we're doing in the literature, or whatever it is. And so that's value added for everyone, because it keeps the science moving the same direction.
And touching a bit on that just with Python, and you asked, or somebody asked about version control as well, you have to think about your Python environment as well, your packages, how it is, how much of that is actually captured in sort of a snapshot. So yeah, it sort of adds some extra burden, although you can do the integration.
Yeah, it's true. I mean, we have some advances for that, but I admit it still requires some work. We have more questions coming up, and this is really great that you guys are being so active. How was your experience to interface KNIME with public websites like the US EPA? Has it the power to read it?
Right, so there it's not direct integration. So how it was published on the ComTox dashboard, it's literally an SFTP link that they provided, and that was downloaded and then sort of loaded into the KNIME workflow. So it's not a direct integration there.
So you're not scraping the dashboard website or something?
No.
You're really just getting the data from EPA and integrated it.
Correct.
And this is maybe a bit more chemical. Do you consider metabolites in your assessments? That is really in the weeds. Daniel, I guess that's for you. Are you considering metabolites?
Well, in general, I mean, always. Like metabolism is like reactions. You know, when it comes to risk assessment, technically we're interested in the actual molecules which come in effect in a biological system. So absolutely.
And then just checking what we are still having as questions. Maybe something about the future. You already said in your last slide you're working on using KNIME in further fields, or to see where it still can leverage your work on something. Do you already have more defined plans where you could implement that, or where you can use KNIME to help toxicologists further do their work?
Short answer is yes, but always as you come to, and I guess that's why with KNIME you can sort of have the fail early principle as well, because an idea comes up, this is the data we have, this is the problem we have, how far can we get, what will be the effort, or do we need more resources to get into that? So we're always looking into trying to do sort of quicker prototypes, proof of concepts as well, and then based on that sort of take it further. So yes, we're always looking at what else we could use it for. But again, that comes, with a support need as well. One shouldn't forget. So, have to always consider that too.
Yeah, and right now we're using it for toxicology, and it's scientific information. So as it becomes more and more efficient and effective at cutting out the need to gather so much information and allowing people to focus on doing their actual job, which is the science and analyzing the science, there will be more people in other parts of the business that will want to use the same thing. Nobody wants to spend all day cutting and pasting. They want to do their actual job.
And when you have the prototype, how long does it take you to actually end up at something that goes into production, that you're also making available for teams or even other teams, other people in the business?
I feel a bit the term is being overused, but it depends. Again, how the workflow is, because KNIME is great with putting components or wrap certain features around, but it depends how the prototype was built. Mm-hmm. You can just take components, because if the workflow is done in a way, as probably many people on the call as well watching the video, is that it can be quite easy. It can take literally a few minutes. But then if it's not done optimally, it can be quite a bit of work.
Maybe it also depends how much support you have from people that understand the subject. And I think we've got to have a shout out again to Soluzione at that point, right?
Yes, that's correct as well, to sort of make sense of potentially crazy ideas. It's like, I see what you're trying to do here, but there's a better way to do this to make it for an easier deployment.
And I think we might have one last question. Could you talk a little about the outputs of KNIME, like formats, report with all the details? I assume that is not a general question, but rather for you, like what's really the output you get from KNIME right now that you're using further? Is it a PDF or is it a table?
I guess anything and everything. So from KNIME tables to PDFs to SDFs to CSV. Again, sorry, going there, it depends on the problem, what is the best sort of format to use. And luckily, KNIME is pretty good with that in terms of providing a good variety of different outputs.
So it is used quite broadly already. This is what I hear from the answer as well. Like, yeah, it's a lot of output. It's not just one workflow. It's quite deeply rooted within your organization. Okay, I am checking my sheet right here, but I don't see any other questions for today. With that, I would really thank you guys for being here. That was a pleasure. And also thanks a lot to our audience. That was a really nice discussion, and you had great questions. And with that, I would wrap up for today. So thanks for being with us. Of course, find us on all the social media, on our website. If you have any questions, reach out to us. You can do that also on the forum. And I think my colleague Heather also has a link somewhere that will come with you in the follow-up email to the forum. So we have a section there. If you have any further questions that we didn't answer today, please go to the KNIME forum, post them there. We will monitor that in the next days and make also sure if there are questions that we are forwarding that to Irene and Daniel as well. And yeah, thanks for being with us today. Have a nice day, a nice evening, depending on where you are. And hopefully see you around soon. Bye-bye.